Welcome, 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 grade 10s to yet another wonderful, exciting life science lesson. Today we're going to do exam revision for paper 2. I know I've said the evil word, I've said exams. I'm afraid exams time is upon us. Hard work, if you've worked hard throughout the year, well done. You're probably going to reap the benefits. It's not too late to do some hard work, right? So what we're going to do today, just going to look at practice questions and kind of see, right, what we've done throughout the year, the kind of questions that we might expect in a test. So the first thing before we get started, make sure that you've got all your resources in front of you, your textbooks, everything there. Some of the questions I'm going to go through with you, and then some of the questions I'm going to obviously ask you to be able to do yourselves. Okay, just a quick overview of what we're going to look at today. Last week, we looked at the cell, and we looked at mitosis, and we looked at the plant tissues and the mammalian tissues. This week, we're going to look at plant organs, right? Remember, I always forget to put the pen on. Okay, we're going to look at plant organs, so you need to know, write all your diagrams about the root, the stem, and the leaf. Right, then we're going to look at how water moves through a plant, and again you got to link it to those diagrams, and we're going to look at certain processes that occur in the plant. There you need to be able to know transpiration is very important there. And then we started looking at the human body, right, the organs that made up the human body, and we looked at the skeleton, and there we also looked at all the different bones. And the most important things there are going to be joints. Okay, so without any further ado, we're going to start with the very first question. I'm going to do the first question with you, and after that, a few of the questions that follow, you're going to have to do yourself. Okay, so let's start off with the question number 1. Now, the first thing we're going to do is, this says, study the diagram below. Study the diagram below and answer the questions that follow. Now, the first thing that we do when we need to look at the diagram is we need to try and identify what are we looking at. And looking at this diagram, the characteristic feature, right, should have been this little feature over here, which is a vascular bundle. But let's have a look and see, right, if the questions might lead us to what is needed from us. Okay, the first one we're going to do is to provide the labels for parts A, C, D, and F. I'm going to put here, we're going to do it on the diagram, right? Then it's much easier for you to see what we have to do. A, C, D, and F. So let's go. We need to do A, we need to C, D, and F. Now, the biggest problem here is if you don't know what you are looking at, you are not going to know what to label. So the first thing, I'm hoping that you were able to see that it is a diagram of a stem. All right, very important. It's a dicotyledonous stem. Why is it not a root? Because a root, right, we see it has a very, right, important central system. And why is it not a stem, a leaf? Because a leaf has a top and it has a bottom. Okay, so let's start. Let's label. Number A is the outermost tissue layer. And the outermost tissue layer is always going to be the epidermis. Okay, that's always a nice given. It's the epidermis. Now we look at C, D, and F. Now, all of these ones have got to do with the vascular bundle. Now, number C, you can see it's got a very thick black outer wall, right, showing that the outer wall is very thick. Almost, there's hardly anything in the middle. Actually, there is nothing in the middle. It's almost, right, the center, the cell wall has taken everything out. And that, I hope you recognize as sclerenchyma. Now, number D and number F. Number F is much easier to see, the reason being that number F has got these nice big hollow cavities, which we recognize as the xylem. Now, if we have a look at number D, when we look at a vascular bundle means that it must have the tissues that are going to transport things. So number D would then be my flow. Okay, so if I don't know what the structure is that I'm looking at, I'm going to really battle to do the labels. And guys, you need to be aware of different ways we can ask the different organs of the root. It can be a line diagram, or over here where we look at cellular detail, right, in quite a lot of detail. Every single diagram, when it comes to a root, a stem, and a leaf, you have to be able to recognize what was the major points. Okay, so what's the next question going on to that? Okay, so there's my diagram again. My next question is, explain how the tissue, explain. Explain means, and look at the mark allocation, that I'm going to need to write a lot. Is tissue labeled F is situated to its function. Now let's go back. What did we say that number F was? And this is a very common question. Number F is the xylem. Okay, so first of all, so we can get our thoughts all into place, the first thing I'm going to do, I'm going to say it's the xylem. Now, what is its function? Its function is to transport water and mineral salts. Okay, up from the roots, up to the leaves. Now, it asked you, explain how the tissue labeled F is suited to its function. So what you have to do here is, you have to tell us what is its best part to enable it to transport, okay, what should I put here? Water. Okay, number one, what makes it so good at transporting water? One of the reasons I could say is hollow. Now I need to explain. It didn't ask me to list, it asked me to explain. Okay, so what does hollow have to do with it being so important? If it's hollow, water can flow easily. Remember, think of a straw, right? So if it's hollow, water can flow easily, or another word we can use is, okay, unimpeded, means without interruption. Okay, I'm putting the points there. I'm not listing them because I'm explaining. What else makes it such a good water transport tissue? Number two is that it's lignified. Right, lignified. And what does that mean? Right, it means that my cell walls are strong. And why do they need to be strong? Right, to be able to take, right, the pressure of the water. Water coming through, right, up the stem at such a pace, if I didn't have the walls, the lignified walls, right, my cell would just crash open. What else could I use? Thin. Right, it's very thin. Can you remember when you did the capillarity experiment? It's thin. If it's thin, water can travel up more easily. Okay, another one. See, sure, it's 5 marks. There's lots that you could have said. So we said xylem, we said transport. Any of the rest of the marks could have come from here. Another one we could have said is that we can put them on top of each other. Put them right on top to form one long tube. Right, and if it's one long tube, then what's going to do? It's much easier for me to transport water. Okay, so guys, when it comes to this question, right, explaining, you actually have to tell me, right, what feature is the reason why it is so good at what it does, and why does it make transporting of water so much easier. Okay, not always, this question is not the easiest one, but a very common question when it comes to asking about xylem. Question number 3, name tissue. So we must name it first and then describe its function. So if we have a look at the mark allocation, we don't want you just to write just a simple thing. Now if we have a look at tissue number E, Tissue number E, I'm going to use a different here, we would find in there. We would find phloem, let me use a different color, phloem over here, and we would find xylem over here. So this red line is a tissue in between the xylem and the phloem. Now, if you think of what do we find in between the xylem and the phloem, we are going to find, right, cambium. Cambium. Right, and that is the name of the tissue. Now, what does cambium do? If you can remember, cambium is meristematic tissue. What does that mean? It can divide by mitosis. And in the case of the stem, what does meristematic tissue do? All right, what does it do? It, all right, plays a role in making more xylem and phloem. And why do we need that? Because in some plants, see, it's worth 3 marks, guys. It's 3 marks. What do we need more xylem and phloem? Secondary thickening. Some plants grow wider, right? They grow wider. We need them. Like, remember our trees, when we looked at our annual rings, what do we see? The plants are going to grow wider, and that cambium is going to, right, give the new xylem and the new phloem so that we can increase in diameter. Okay, we're not finished with this question yet. Right, now this is quite an in-depth question, but it also, one of the things that you need to be able to do is to compare the different kinds of tissues to each other. Now remember, why are we comparing the different types of tissues? Different tissues have different structures depending on their function. Okay, so if we look at their function, or if we look at their structure, we can sometimes see what either their function might be, or looking at their function can see what their structure is going to be. Now, we have to compare tissue B, compare. Now you can see here, comparing is in a table, tissue B and tissue G. Now guys, if you do all the adding up, do you see how important it is to be able to label your diagrams? Because number B is here and number G is over here. Now, if you were not able, from the beginning, if you were not able to identify that it was a dicotyledon in a stem, how many marks would you have lost? A lot of marks. Because if you are not able to put labels to a diagram, then you're not going to be able to answer the questions. So it's really important that you know your diagrams and you know your labels very, very well. Okay, so what have we got? Number B is, and you can see it's stained slightly darker, not as much as number C, which we said was sclerenchyma. Right, and number B, I hope you guys all got it, is parenchyma. Number G, thin-walled, there might be spaces, looks like a vacuole's taking over, all right, and it's also the most abundant tissue is my parenchyma. Now, see how important the labels are? Now, if you got this wrong, 10 marks for the label, just for the table. Okay, so we don't want to do that. So let's put in our labels over here. Our tissue B is parenchyma, and our tissue D is parenchyma. Oh, and I don't even read my table properly because that must be in the first lab over there. Dead or alive, both of them are alive, because which one would be dead? That would be, right, that would be sclerenchyma. Sclerenchyma, when we get to our sclerids, our stone cells, the cell wall takes over the whole cell that it actually dies. It's like the xylem as well. Primary cell wall present. Yes, all right, both of them. Primary cell wall present, yes, for the parenchyma. It's actually thickened. Is a primary cell wall present in parenchyma? Yes, even though it is much thinner. Okay, now we get to the function. What is the function of parenchyma? Now, if we go back to our diagram, parenchyma is usually found right underneath the surface of the stem. So that tells us something about it, right? And the fact that it's also got these thickened cell walls. So what is the function? Here we have a strengthening function, right? And we actually use the word mechanical support. Whereas my parenchyma, right, if we have a look there, very thin walls, right, a lot of it. So we can see that nice large vacuole, it's usually right for storage. So description of cells, putting all of this into the description. My parenchyma, strengthened, okay, oopsie, not spot, at corners. That's very important. All right, the corners. Let me write that better. Let me do that better. Okay, corners. Okay, corners. There we go. Here, what do we say? Thin walls and large vacuoles. Now remember, let's relate structure back to function. Okay guys, time for a quick break. Go and stretch your legs, right, and have a little bit of water or something. We're going to be right back because you guys are going to be doing the next question. Welcome back. Hope you had a bit of a stretch, get those legs going, get all the blood flowing. Right, now this question is up to you. Right, the next question, I'm going to read it through with you, but then you are going to answer the questions that are relating to it. Okay, you have to examine the photograph below of a transverse section through a root, right, of a mealy plant as seen using a light microscope and answer the questions below. Okay, I'm going to give you 2 minutes. That's all you're going to get. Right, remember, already they're telling you that it's a root. So you should be able to know your diagram. You must know the structure and the labels of the different areas. I'm going to give you 2 minutes. I'm going to, all right, put the picture up, then I'm going to put the answer up, then I'm going to put the picture up again. Okay, so for you so you can see, answer the questions, look at the diagram, look at the picture of the cells, all of those things again. So 2 minutes, right, your time starts now. Okay guys, how did you do? I hope you were able to see, right, leading from our last question, you should have noticed that there were some similarities in the questions that you were asked. That sometimes there's certain characteristics or certain questions that become more oftenly asked or more obvious than others. Okay, so first of all, we said to you that it's a section of a root. If I didn't tell you it was a root, right, what you should have been able to see, and that's one of the questions, C would have been able to give you your function, okay, that it is a root, because they haven't shown you all the inner part of the root. So you might have thought, oh, I'm not quite sure what this is, but that little structure over there was your hint. Now, the first question was quite long, right, because it involved a few multiple incidents of the question. Okay, and the first one is name the tissue types. Okay, now it's quite important, because when we look at the diagram of the root, so for example, over here, okay, this area over here, they've asked you for the type of tissue. Another kind of question could be, what is this area called? And that would actually be something totally different. So when you read your questions, sometimes in the wording, these little subtle differences that you might need to pick up on, right, about what are they asking you. Here they're asking you the tissue type, what type of tissue, not what is that area called in the cell. Now, leading from our last question, the tissue types, all right, we did in the last question as well. So what did we notice over here? Number A of thin walls, right, and they have very large vacuoles in them. Whereas if we look at number B, although not characteristically found in a root, the question has said that these cells, right, are in the root. What do we notice? We notice that they have vacuoles, but what do we see about the cell walls? We see that the cell walls, right, are thicker over here in this area than those ones. And if we look at it, we can actually see that there's living contents inside the cell. It hasn't taken over the whole cell. So we know that this is parenchyma, which we have done previously. That's why I said you could do this. You guys must do it yourself, right? Hoping the first question led you a little bit to this one, which then makes this nice thin-walled cells parenchyma. Okay, but that's not what the question asked us all. It is for 6 marks. You had to do a few things. Number one, name the tissue type. So let's start. Number one, what is A? A, we go back here. What is A? A is parenchyma. Okay, so there we got. First of all, we named it. Then it says, for each tissue, state one distinctive feature. Okay, so you had to say, why? One thing. Why did you see that as parenchyma? And if we go back to the picture, what do we see? My first thought always is the thin walls. My first thought, whenever I think of, right, the parenchyma, I think of these very thin walls. I think of the very large vacuoles. Okay, so that for me would be the most obvious. It says state one distinctive feature. So you had to use the picture on the diagram, right? You can't think to yourself, no, teacher gave us another one. You must use what information do you see in front of you. Okay, so our parenchyma, 1 mark. What do you think its distinctive feature is? Thin walls. Some of you might have said the large vacuole. And some of you might have also said that there was intercellular spaces. Any of those, right, are correct in identifying it as parenchyma. Then, function. So that was number 2, one distinctive characteristic, and the last one, number 3, what is its function? So I've got parenchyma, yes, number 1. Those are the distinctive features, number 2. What then do I need to do? What is its function? Storage, right, of minerals. What else? It helps with the turgidity of the cell to keep the plant upright. There we go. I have my 3 marks that I needed for parenchyma. Now I need to do the same for parenchyma, right? So what do? I write. Okay, so what is number B? It's parenchyma. What is its distinctive function? Thickened cell wall corners. Okay, so there's my 1 mark. There's my 2 mark. What is its functioning? Mechanical support. And, all right, to keep strengthening. It's a strengthening tissue. There we go, my 6 marks. A bit similar, all right, a different structure. One was the stem previously, one was the root, but again, look at the cells. Why is this one made like that? Why is that one made like that? Okay, and then last question there, state the function of cell type C. Oops, wrong way. Cell type C was your root hair. Right, was your root hair. And what is the function of the root hair? The absorption. It just asked you for the function. You didn't have to go into too much detail. The function of it was to absorb water and mineral salts. Okay, right, now our next question. Our next question, as you can see, right, is study the graph below, which shows the rate of transpiration. Guys, as soon as you see certain words, all right, certain processes need to, certain kind of thought process need to start going through. Transpiration. What is transpiration? When I lose water in the form of water vapor, usually from the leaves, from aerial parts of a plant. So what am I doing during transpiration? I am losing water. Where do I lose water from? From stomata. What happens? It usually happens during the day when the stomata are open. At night, the stomata are closed. Those are the kind of thoughts that need to start rolling around in your head when you think about the rate. Because remember, the rate was speed. Also, I need to think of what things will affect how fast transpiration occurs or how slowly. Okay, so let's have a look at the graph. Okay, study the graph below, which shows the rate of transpiration. Now, this is where you need to look very carefully. The rate of transpiration is this blue line. Okay, there we go. Now, right, the pink line, oopsie, let's start with pink, shows the rate of water absorption. So in other words, what you're looking at is a graph showing you how a plant loses water and how a plant absorbs water. Let's have a look at our questions and see where they are going. Okay, there are 4 questions here. The first one, name 3, remember there's always a reason why it's in capital letters. They only want you to state 3. If you do more, not going to look in it, which could directly influence the rate of transpiration from the leaves of a plant. Now, if you notice in this question, they haven't said to you what will cause the rate of transpiration to increase, what will cause it to decrease. What they've said is, name 3 environmental factors that could influence the rate of transpiration here. So you don't have to go and be more specific. You can be more general. Okay, so let's answer the first one. The 3 environmental factors. They're actually 4. Okay, but you need to, some of you will think of a different 3. The 3 environmental factors, can you remember what they were? Wind will influence the rate of transpiration. Light intensity, temperature, and humidity. Okay, so they say would directly influence. That means they will have an effect. Now, question number 2 says, choose one of the factors that you have mentioned and explain why it could cause the rapid decrease in the rate of transpiration after 4 o'clock. Let's have a look here. Have a look at my 4 o'clock is 1600 hours. So let's go up here. Which factor decreased? Now, there can actually really be only one answer here. Because if you notice, we are now talking about a specific time of day. Okay? You can maybe talk about 2, but let's start off with the most logical one. Okay, now let's think about it. The question they said was, from your 4 answers at the top, here you gave 4 answers. Which one of those would cause the rapid decrease in the rate of transpiration after 4 o'clock? Okay, now being winter time at the moment, right, and I notice you guys when you're traveling home or when maybe you're outside playing, what is one of the first things that you might have noticed? That it gets darker a lot quicker. So let's have a look here. Why do you think at 4 o'clock, what happens, you think, when it gets darker? This is going to be my first one. It's going to be my first option. I'm going to choose light intensity. And why does it cause a decrease? Because as it gets darker, what happens to my stomata? My stomata close. And when my stomata close, what's going to happen? My rate of transpiration is going to decrease. Okay, it's going to lower down because what's happened? No more, it's dark, no more light. My stomata is going to close. Right, question number 3. Describe what the plant might look like at 2 o'clock and explain why. Have a look here. Have a look at 2 o'clock. Right, let's go up again. Have a look here. When we draw this, let's put our 2 rates of transpiration, rate of water absorption. Oopsie, let's go there. Okay, have a look here, guys, at 2 o'clock. What do we notice over here? What do we notice? The plant is losing more water than it is absorbing. Okay, so now if that is the case, let's go and answer. So if you were to answer your question, okay, what do you think? More water is going out than coming in. So what might happen to your plant? Okay, here's a word. They might wilt, right? They might start to droop a bit. And why is that? And there's your reasoning from the graph. Okay, it might start to wilt because it's losing more water than it is absorbing. Okay, use your graph, write the details on your graph to be able to do that. Now the last one, question number 4, give a reason why the rate of water absorption started to decrease in the late afternoon. Look here, let's go back. Okay, let's go and have a look. Why did water start to decrease in the afternoon? Right, because what happened? Right, what happened? Late in the afternoon, water absorption. Okay, why do we not need to absorb more water? Because it also gets cooler. And when it gets cooler, what else happens? And it's darker. The stomata close. So we decrease the rate of transpiration. Right, so we don't need to have such a long right? So we have less transpirational pull, and therefore we need less water. Okay, guys, time for a break. And after that, we're going to come back to our last few questions. All right, quick stretch, and we'll see you right now. Okay, welcome back again. I always hope that you stretch, get everything going. Okay, our last section, we're going to start. Almost finished. I know some of you are so tired. All you hear is exams, exams, exams. Right, be careful. Okay, guys, don't get into that trap so early. Make sure you're eating properly. Make sure that you're getting enough sleep. Right. Okay, the next question comes from our skeleton section. Now, study the diagram below, which shows a section through the arm. Once again, telling us the arm and the elbow joint of a human, right? So we can see, again, here we got a whole lot of labels. So the first thing it should be going through your mind, okay, I'm probably going to have to label, right, these different bones. And it tells me that here is a joint where two bones meet. What kind of questions can we ask over here? Okay, the questions asked, right, question number 1. Label structures B, C, and D respectively. I'm going to do each of them like that, then as much. Okay, so we're going to do it on the diagram. It's always much easier to relate the structure, all right, when we see the diagram. B, C, and D. So let's highlight. We want B, we want C, and we want D. Now this is quite, C and D I can see is could be quite problematic for us. Now, number B, if you have a look at number B goes from here to here. Okay, now what is that? That is a bone joining with a bone. So number B from our mammalian tissue, right, even if we haven't done, we might not remember the joints, a structure joining bone to bone is called a ligament. Right, now if we have a look at C and D. C and D, this number A, right, would probably, it's the elbow, would be the humerus. Now if we have a look at C and D, how do I remember? How do I remember which one is which? Now I always remember, right, on the outside, if we go over here, right, the radius always is the bigger one, and that goes to your thumb, and the smaller one on the inside over here is your ulna. So if we have a look over here, the bigger one, right, is your radius, because why? It's on the outside. Here's my elbow on the outside, and then number C is going to be my ulna. Okay, that is quite a challenging one, but you need to always remember which one is that. Question number 2, identify the type of synovial joint. Now, if we can remember, you need to know your joints, you need to know the different types of joints, and an elbow is an example of a hinge joint. Okay, because it can only move, it can flex or it can extend. Now, question number 3. State 2 functions of the capsule. Now, if you have a look here, right, here is the capsule, and it's a capsule membrane. Now, what is the function of the capsule? Number 1, it needs to make fluid. That's very important. It needs to make synovial fluid. And why is that? For lubrication. So joints can move easily. Right, another one of the capsule is to keep the synovial fluid in place. If you didn't have a capsule, you'll have like your knee or this elbow would be swollen all the time, right? Because then it would just go out, it would fill up all the spaces, and here we just want to keep it contained, is to keep the synovial fluid contained. Right, so what do we have a capsule? Right, so that we can produce synovial fluid, and also that we have, right, an area, okay, where we have movement. Isn't that the whole thing about a synovial joint? Right, synovial joints allow movement. Remember, we did cartilaginous joints and we did fibrous joints, but very little to no movement were allowed. So with having a capsule, if we go back to our diagram here, where the two bones meet, we have this space, and in that space, right, we have the fluid. And what else do we have? We have movement, because that elbow is going to move up and down. So that capsule helps us with doing that. All right, and our question is, our last question on that, right, one function of B. And what was number B? Aha, B was a ligament. And we know that a ligament joins bone to bone. Okay, now, very interesting one. Question number 4. Name the structure that will attach the muscle to the area of the bone marked X. Okay, so what we're going to find over here, we're going to have a structure over here that comes here. And then what we have is a muscle, which is a muscle. And a muscle doesn't join directly to bone. What does a muscle do? A muscle, that structure number X, is that little structure that is going to join a muscle to a bone. All right? And what do we call the structure that joins muscle to a bone? It is a tendon. Yes, yeah, I'm changing colors like a rainbow here. Right, then question 5. Guys, you need to be able, right, to make certain kinds of drawings, to be able to make simple drawings. You need to be able to prepare to do that. Now, if we have a look, question number 5, have a look here. They want you to make, here's a bone, all right, those are all bones, and they're long bones. And for 5 marks, they want you, right, to make a labeled drawing of a longitudinal section. Longitudinal, long, not across, right, long. Long means from top to bottom. Okay, so here goes. My drawing skills are not awesome, but what do we notice? Okay, I'm going to do more space there. Okay, we're going to have that. Okay, so when I draw a bone, I notice that it has ends. And those ends are called epiphyses. Epiphysis. All right, epiphyses. Okay, then they have a long portion, all right, called the diaphysis. So we have the ends. Okay, then what do we have? Usually around the ends of long bones, what do I have? Now remember, you guys need to do a biological diagram. So I don't have space here. Well, let me make it. Let me try. There we go. Hyaline cartilage. Right. A longitudinal section through a long bone. Okay, there's my heading. Here's my diagram. So one for my heading, one for bone, probably the rest for labels. What can I label the bone here? In the ends, the epiphyses, what do I have? I have spongy bone. What do I have in my diaphysis? Compact bone. And what do I need to add to my diagram? I need to have my bone marrow, right, or my medullary cavity, right, I'm going to put there bone marrow running through the center of my bone. Now, if you have a look at that, right, you don't have to be a really, really good artist to be able to draw that bone. All right, so what you probably find, 1 mark for your heading, 1 full mark for your diagram, and it's out of 6, 4 marks for the correct, any of those correct labels. Not a very complicated diagram, quite easy to do, right? You just need to obviously remember the labels. Okay, we're not going to have too much time for this, But one of the things that you guys need to be able to start to do is at the end of a paper, right? And this I sometimes think is the part of the paper that you stress about the most, is you have got an essay type question where you are given a topic, for example, the one in front of us here, and you have to write something that has 17 facts or marks worth of facts. And you've got to write it. It needs to be, all right, written properly. You shouldn't be using bullet points. But sometimes, if some of you battle, you find that maybe writing in bullets might help you, right, just to get your thought processes in place. And that last question at +3 basically is, how did you write your essay? And did you write biologically, or were you able to understand the terms that you used? We call that synthesis. Right? We call that synthesis. And you'll see your teacher has a little rubric, and that rubric tells her, right, all about the points that you needed to do. Now, guys, when you look at this, right, break the question down part by part. And each part, you can maybe make a paragraph of that. And be careful, it doesn't have to be pages and pages and pages, right? It's a biological essay. It's about points, getting your points across quickly. It's not an English or a language or a thing where you have to all speak all about emotions and things like that. Okay, so let's have a look. If you were to write the essay, how would you plan it? Okay, let's have a look at the question. It says, describe the absorption of water by a root hair of a plant. Okay, so what's one of the things I'm going to do? I'm going to describe how water moves into the plant. Okay, so I'm going to make a little paragraph there. Then it said, as well as the lateral movement of the water from the root hair to the xylem of the root. Okay, so water moves into the plant. I'm going to describe all about the root hair. And I'm also going to say how the root hair is adapted to its function. Okay, then now, see, remember guys, this is not your essay, this is just your points across. What's the next thing I'm going to say? I'm going to talk about how it goes from the root hair, planet, through the epidermis, which is that, through the cortex of the root, through the endodermis, you see, I'm putting words down, till it gets to the xylem. Right, then the last questions. Explain the different ways in which the xylem is structurally suited to transport water up the plant. All right, and that is a nice easy one, because why? We did it in a previous question when we explained how it was suitably adapted. Okay, guys, that's all from me. I wish you all the best for the exams, right? Well, I'm hoping that you do very well and you do your best. You put all the work in. Until next time, cheerio, bye.